Hello, and welcome to the Clinical Care Options Infectious Disease Podcast. I'm your host, Tiffany Hensley McBain. Today's episode is part 16 of an ongoing series entitled COVID-19 Answering the Questions. This series features brief updates on the latest incidents and clinical data related to COVID-19, diagnosis, prevention, and management, each followed by an in-depth question and answer session designed to address infectious disease specialists' most pressing COVID-19 questions. During this episode, Professor Sharon Lewin from Melbourne, Australia, will provide an update on immunity to emerging SARS-CoV-2 variants and implications for vaccination. For more information about Professor Lewin and for a link to additional online education from CCO's COVID-19 Comprehensive Resource Center, please visit the show notes for this episode. Now let's get started and hear a COVID-19 update and answers to clinician questions by Professor Lewin.
Thanks very much, Tiffany, and a great pleasure to be presenting to you today. As usual, we'll start with a global update on COVID-19, which continues to spread greatly around the world. This shows you the latest data from the Johns Hopkins Global COVID-19 dashboard, with over 100 million confirmed cases globally now, over 26 million of those in the US, followed by India, Brazil, United Kingdom, and Russia. We also see incredible impact on deaths, with over 2 million deaths and over 400,000 of these are from the US. So COVID-19 continues to ravage the world, and urgent solutions are certainly required. So today, the focus of the talk is on a very topical area on vaccine or infection-induced immunity to emerging SARS-CoV-2 variants that many of you will be hearing a lot about. So first of all, for the non-virologists, what do we mean by a genome mutation or a variant? Well, on the left of this slide is a schematic of what the SARS-CoV-2 virion looks like. In the inner part of the diagram, a coated virus with spike proteins surrounding the outside of the cell, forming the classic corona, which defines a coronavirus. Around that schematic diagram of the virus are the major proteins that are encoded by the virus in order to allow it to replicate. And you can see those proteins detailed in different colours. In the orange colour, you'll see the spike protein, which is the genetic code for the protein that protrudes from the surface of the virion. On the right is a schematic diagram of the mutations we know are associated with the variant B1.1.7, which was originally described in the United Kingdom, but now found in many parts of the world. And you can see lines drawing through these proteins, showing the location of mutations in the B1.7 mutant. And you can see that the mutations actually extend into many proteins, not just spike, but there's a large amount of mutations sitting in the spike protein gene. There's a combination of mutations, which are shown there in a teal colour, but there's also some deletions shown in a grey colour. It's just a schematic diagram to make you aware of where these mutations are. They're in many different proteins, predominantly in the spike protein, and they consist of substitutions as well as deletions. And in the lower part of this diagram is a more detailed schematic of this spike protein gene. And there's lots of different acronyms here, but I just want to highlight a few things on that gene. First of all, you'll see that there in the middle is an arrow saying S1, S2, and that shows the division between two parts of the spike protein, commonly called S1 and S2, which requires to be cleaved in order for the virus to enter. You can see a green section, which is the RBD, the receptor binding domain, and a blue section to the left, the NTD, the N-terminal domain. And I just want to highlight those two areas because they're often the site of the mutations that we worry about the most. Now on the next slide, you'll see a 3D illustration of what the spike protein looks like. Now this is in a red and maroon colour, so it's a slightly difficult thing to view in these colours, but on the far left is the native spike protein, which consists of three proteins together forming what's called a trimer. The upper part of the trimer is the part that binds to the cell, and the lower part is the part that's attached to the virus. And this is just an example so you have a visual sense of what these mutations are and where they're located in a three-dimensional form. And we've just highlighted one mutation, quite an important mutation because it's present in most of the mutants that have recently been described, the N501Y mutation that sits in the RBD, or the receptor binding domain. Now, this mutation of itself is really important because as it's in the RBD, it affects binding to the receptor of SARS-CoV-2. Many of you will know that that receptor is the ACE2 receptor. And it's shown to you on the far right, the trimer on the bottom, the red and maroon, binding to the ACE2 receptor, it's shown in the light blue. And what a mutation such as N501Y does is it alters the conformation of the receptor binding domain, allowing it to actually bind to ACE2 tighter than the wild type. And this allows it to give, we think, more efficient entry and therefore ability to replicate at higher levels. Now, on the next slide, you can see another important role for the RBD, or the receptor binding domain, as well as the NTD, or the N-terminal domain. These sites in the viral S protein are also important for antibody binding and therefore neutralization. And although this is shown to you in slightly different sort of schematic, on the left in yellow is a representation of what the spike protein looks like, or specifically the RBD, the receptor binding domain, and a few mutations highlighted on the RBD. And you can see the interaction with certain monoclonal antibodies. And this can be modelled quite nicely using highly sophisticated imaging tools. And on the right shows you the NTD now, just the N-terminal domain, interacting with another different antibody. So antibodies can bind to lots of different parts of the spike trimer, not just the receptor binding domain. So having seen these schematic diagrams, I hope you've got a good appreciation for the fact that mutations will affect binding to the receptor and therefore ability to infect a cell, and will also directly infect how the virus interacts with antibodies, either naturally induced antibodies, vaccine-induced antibodies, or manufactured monoclonal antibodies. Now, what we're worried about most with the vaccine is how these mutations might affect neutralization. And I want to spend a moment describing how we measure this, because it can be a bit confusing when you read the literature. So neutralization of viruses can be quantified in vitro using different assays. When assessing neutralization, you assess the antibody or the vaccinee's plasma against the laboratory strain of the virus. And the viruses can either be the complete original virus, the complete virus, which is the original virus, or what we call wild type, or virus that contains mutations, or scientists will assess the activity against pseudoviruses. And pseudoviruses mean there's a common virus backbone, and you've got either the whole spike protein inserted into that virus, either the original or a mutant, or you can actually generate just a specific single mutation at a specific site, or a combination of mutations. And it's important to distinguish between these two different methods of assessment of effects of virus mutation. Now, the results can either be reported as the concentration of the antibody needed to suppress virus replication, and therefore a higher number will mean that it's a more resistant virus, or it can be expressed as dilution of an antibody that blocked at least 50% or 90% replication of the virus. And because it's a dilution, a higher number means higher potency of the antibody. And this can cause a little bit of confusion. So I just wanted to explain those two readouts so that you can interpret the literature. All right, so what are these variants that we're most concerned about? Well, the first one was described in the United Kingdom. It's labelled B117. It contains 23 different mutations, as I've already shown you, including 8 mutations or deletions in the spike protein. The N501Y mutation, which we've already discussed, sits in the spike RBD, receptor binding domain, and alters the affinity to human ACE2. I've already shown that to you schematically. We then subsequently heard about the B13.351, originally called the 501Y.V2 from South Africa. This also has 8 defining mutations in the spike protein. Some of them are similar to the UK strain or 117, but others are different. And in the B1.351 strain, I hesitate to call it the South African strain because we know that this variant is in many countries, so it shouldn't be used to stigmatise South Africa. But it's a bit of a mouthful, the B1.351. There's 3 residues in the RBD, which we think are of importance, the E484K, 41K, 417N or T, and again, that same mutation, the N501Y. And then more recently, we've heard about another mutation, P1, from Manaus in Brazil. This has 17 defining mutations, including the same 3 mutations in the RBD in the South African variant. Now, we've already getting population-based data demonstrating that both the 1.7 and 1.351 strains appear to be more transmissible. And this has been demonstrated at a population level. The transmissibility of P1 is unknown, but based on the kinetics of change of infection in Manaus, Brazil, it's thought to also affect infectiousness. Now, recently, the Public Health England used a matched cohort analysis to look at whether the mutant 1.7 actually alters the risk of death. And there's a suspicion or a suggestion that there is an increased risk of death of 1.65 for individuals infected with the B117 variant compared to non-B117 variants. And this is important to note that these are still not reviewed, and also there are additional unpublished analyses demonstrating increased risk of death. Although there's an increased risk of death, one has to balance this with, of course, the absolute risk of death and the effects of this increased impact. But these are certainly variants we are worried about because of their effects on transmission and appears the effects on disease severity as well. So the first studies that looked at what happens to neutralisation of these variants were assessed using convalescent plasma. This is a publication that comes from South Africa that assessed the neutralisation of live virus. So this is not a pseudotype virus, this is live virus, the B1351 virus, grown from a sample collected in South Africa in July 2020 and tested against sample plasma from people who were infected with a non-mutant variant. And on the right are the IC50 values, the concentration of antibody allowing for a suppression of 50% of viral replication. And this shows you the results of 6 donors, each row a separate donor. And it shows you in a nice color-coded manner the fold increase of antibody required to suppress replication by 50%. So therefore, a number above 1 tells you that that virus is more resistant to the antibody, meaning that it needs more antibody there to suppress it. And you can see there's quite a wide range here with an isolate of with the B1351 tested against plasma collected from participant 2, and you can see that 200 times that the antibody level required to suppress replication of the mutant virus was 200 times compared to what was required in the wild type. So this would certainly suggest that you would have reduced natural infection to prevent you from reinfection with this mutant strain. Now, the next, this is another similar sorts of study that assessed neutralisation of the wild-type virus, a virus that contains the RBD only of B1351, and the B1351 virus tested against a range of serum samples from individuals previously infected with SARS-CoV-2. And here, this is a slightly different way of representing the data. It's the dilution of antibody required to suppress replication. So a higher number means that the antibody, that the virus is less resistant. So on this diagram, you can see that if you looked at the titer of less than 25 or greater than 400, if you just look at the extremes, in the original virus, meaning the wild type virus, when you test it across the serum samples collected, about 50% have a very sensitive to the virus and about 50% intermediate sensitivity. If you look then to the far right, which is the variant, the B1351 or the mutant virus, you can now see that almost 50% of plasma has greatly reduced sensitivity to neutralisation with an ID50 titer of less than 25. Less than a lower value means more resistant. And so you can therefore see that the mean ID50 titer for individuals with mild to moderate disease against what, oh sorry, I didn't explain that. These samples were collected from individuals with mild to moderate disease or severe disease. When the investigators looked at the mean ID50 titer for individuals with mild to moderate disease against wild type, it was 488. The mean titer for individuals with severe disease against wild type was 4,212. We know that with severe disease, you tend to make more potent and high levels of antibodies, and that 48% of individuals had no detectable neutralisation against the mutant virus. Now we move on to a series of publications that have looked at the effect of mRNA vaccine elicited sera against these same mutant viruses. And these studies have been done in participants who have either had the Pfizer vaccine or the Moderna vaccine or both. So in this study, the investigators assessed neutralisation of SARS-CoV-2 spiked HIV-1 pseudoviruses by sera from participants who received either the Pfizer vaccine or the Moderna vaccine. And the average time that samples were collected was about 8 weeks after the second vaccine. Here they used pseudoviruses, so they could test each specific mutation. Here, the 3 mutations in the receptor binding domain or the RBD, or the mutations together, and what effect that had on neutralisation. So this is an assay called the PVNT, a neutralisation titer. It's a dilution assay. So the higher the value, that means it's more the virus is more sensitive to that it means that the sera is more potent against that particular virus. So if you look on the left, whether you had wild type virus tested here or a mutant, this particular mutant at position 417, there was no difference in the titer required to neutralise. If you look at the far right, when you have the combination of the mutations, you can see that the dilution is reduced. It means that the antibody is, that this mutant virus is more resistant to the antibodies being tested, significantly different from the effect of that same plasma to wild type. So that says that the vaccine-induced sera requires a higher level in order to neutralise the mutant virus. This subsequent study also used the same, a similar approach, but here they only assessed the vaccine-elicited sera from recipients of the Moderna vaccine. They assessed the neutralisation again of SARS-CoV-2 pseudovirus by sera from both non-human primates and people who had received the Moderna vaccine. The samples here were collected 1 week after the second dose. And here they were able to actually quantify the effect, and they said that they quantified the effect when they looked at the pseudoviruses that contained either the UK variant. Now I'm now looking at the, showing you this in the figure on the right, the UK variant B117 spike protein, or the South African variant B1351 spike protein, you can see there's a greater effect on neutralization of the B1351 sample, and estimated to be a 6-fold reduction in ability of sera to neutralize that mutant. And all samples, either from non-human primate and humans, fully neutralised all variants with titers above those found to be protective in non-human primate challenge studies. So that's important. We can say that there is greater concentration of antibody needed to neutralise the mutant virus, but the concentration achieved in vivo in both non-human primates and humans is achievable, which means that this may not necessarily translate to an impact on efficacy of the vaccine. We don't yet have the results of that with the RNA vaccines. This next study looks at neutralization again of pseudoviruses this time, and looking at the Pfizer elicited sera. It assessed neutralization of both the UK variant B117 and wild type, or here called the Wuhan lineage virus, in a different type of pseudovirus, looking at sera from participants who had received the mRNA vaccine, and they assessed the activity of that sera 21 days after the second dose of the vaccine. And you can see on the right is the neutralization titer, and this is a dilution again, but you can basically see that the titer is equivalent, meaning that the plasma neutralized both the wild type strain, here called Wuhan, or the UK strain, here B117, with similar concentrations of virus. Another study, again, looking at the Pfizer vaccine, this time looking at both variants, the B117 and the B1351 study. Samples collected 2 and 4 weeks following the second dose. And a calculation here of the ratio of 50% neutralization of the mean titer of the B117 versus wild type, or the B1351 versus wild type. And here you can see that there is no effect of the neutralization of the B117 with a ratio of 1.41, but a reduced activity with the South African variant, or B1351, shown here on the far right diagram, showing you the results 2 weeks post-vaccination as triangles and 4 weeks post-vaccination as circles. So, what are the potential implications for the efficacy of an mRNA vaccine? Well, with the variant that was initially described in the UK, the B117 variant, there seems to be no significant impact on the neutralization activity of sera collected from recipients of either mRNA vaccine. Now, ex vivo, when sera from the Moderna vaccine was tested against the B1351 variant initially identified in South Africa, there was a 6.4-fold reduction in neutralization activity. And it's important to realise, and this quote comes from one of the papers I've shown you, that despite this reduction, the neutralising titer levels with 501Y.V2, which is the B1351 variant, remain above levels that are expected to be protective based on challenge studies in non-human primates. There are now plans to test an additional booster dose with the Moderna vaccine to boost neutralising titers, and also plans to develop a different construct that actually adjusts for the variant, particularly the B1351 variant. Now, in the assessment of the Pfizer vaccine, again, this was done in separate studies with a separate sort of technology against the South Africa variant initially identified in South Africa. There was a 0.81-fold reduction in neutralization activity. So the impact on mRNA vaccine efficacy in the real-world setting, meaning assessing the vaccine efficacy in an environment where there's high levels of the South African variant, we don't yet have that data. We can show ex vivo a reduced neutralization, but we don't yet know if that translates to reduced efficacy. These are both press releases from the Johnson & Johnson and Novavax. They're not preprints or publications as yet. But just very briefly, the Johnson & Johnson vaccine, which you will know was a single-dose vaccine using an adenovirus 26 construct, presenting the spike protein with a double proline mutation, was tested on 48,000 people, a randomized placebo-controlled trial with a primary endpoint of preventing moderate to severe COVID at 28 days post-vaccination. Study was done in a number of settings, I think US, Latin America, and South Africa, and the efficacy overall was 66%. However, in the US, it was as high as 72%, where these variants we think were uncommon. But in South Africa, where the variants were common, where 95% of COVID cases were due to the B1351 variant, the vaccine efficacy was reduced. It wasn't eliminated, it was reduced to 57%. Overall, 85% effective in preventing severe disease, which is really what we're trying to achieve from our vaccine implementation. And there was complete protection against hospitalisation and death for all variants, which is encouraging. Now, the Novavax vaccine had a phase 3 trial in the UK and a phase 2B trial in South Africa. Protein-based study with over the vaccine is a protein-based vaccine with a specific adjuvant called matrix M1 or M1, and includes the spike protein with the 2 proline mutation. And the primary endpoint here was preventing symptomatic COVID at 7 days post-vaccination, so important difference, different primary endpoint. Overall, efficacy was 89%, and more than 50% of cases were due to the B117 variant in the UK trial. In the South African study, more than 4,000 people, so much smaller study. It's important to be mindful of that. They assessed efficacy overall, and then excluding people living with HIV who participated in the study. And in persons without HIV, the vaccine efficacy was 60%, and overall, which included people living with HIV, the vaccine efficacy was again around 50%. So, and here 92%, similar levels to the Johnson & Johnson vaccine, had a confirmed case with B1351. And I will end there, and we have time for questions. Thanks very much.
Thank you very much, Professor Lewin, for that great overview. But we do have quite a few questions, and we'll do our best to get to as many as we can. Siskin asks, do you think a booster dose of Pfizer or Moderna are indicated? And if so, when?
At the moment, we have no evidence to support that. And at the moment, the recommendations are the Pfizer as given as 2 doses 3 weeks apart, and Moderna as 2 doses given 4 weeks apart. We have no evidence around booster doses, if they will be needed, what they will do, and whether they will enhance protection against these mutant viruses. But that will be an important question to answer in future clinical trials.
Okay, and Mayer asks, is the speed at which COVID-19 is mutating faster than would normally be expected? How does it compare to the mutation speed of other viruses such as influenza?
We have a very good understanding of the mutation frequency of coronaviruses and particularly SARS-CoV-2. It has a lower mutation rate than influenza and than HIV. The reason why we're seeing mutations evolving quickly is because the numbers of people that are being infected. If you take a virus in a laboratory, for example, and you passage it, meaning that you grow it, the virus, take off the supernate and put it again on new cells and passage the virus. The more you passage a virus, or the more people it will infect, the greater chance it has to have a mutation. It's an RNA virus. Mutations occur. They occur at lower frequency than influenza, but when you get so many people infected, it has a lot of opportunities to acquire mutations. And then if there's any advantage to that mutation, such as enhanced transmissibility, the mutant will then become the dominant form.
Okay, and then this is a follow-up to that one. What type of interventions can you recommend to slow down the mutation rate?
The most important thing to slow down the appearance of mutations is to stop transmission. And that is why non-pharmaceutical interventions and eventually vaccines, should they reduce transmissibility, and there's certainly emerging evidence that we will see that the vaccines have an effect on transmissibility, stopping the number of new people infected will slow down the appearance of these mutants.
Okay, and Caroline asks, based on this info, what can I tell my patients that have been vaccinated twice, so they've received both doses, in terms of their ability to see their grandchildren or get back to some kind of normalcy?
There's a few answers to that question. The first point of the vaccination is that it reduces severe disease, symptomatic disease, and hospitalisation. And so therefore, the chance of becoming very sick or even moderately sick with coronavirus will be greatly reduced by both RNA vaccines by 95%. There is still recommendation to maintain non-pharmaceutical interventions such as mask wearing, physical distancing, hand washing at the moment, even for vaccine recipients, because of the unknown of whether vaccine recipients can still carry the virus and transmit it to others.
Okay. And Nathaniel asks, what is the recommended time frame between COVID-infected patients and vaccination? So if they previously had COVID, when should they get the vaccine?
Yeah, the current recommendations in the US are that you shouldn't vaccinate someone when they're symptomatic or unwell, and after resolution of all symptoms and out of isolation, which is usually 10 days for someone that has COVID, it's appropriate for vaccination. The only time when you would delay vaccination is if the patient received either convalescent plasma or a monoclonal antibody, and the recommendation is to wait 90 days because there's a possibility that the convalescent plasma or monoclonal antibody will reduce the efficacy of the vaccine.
Okay, and LM asks an interesting question. Are field efficacy estimates going to be adjusted for saturation of healthcare delivery capacity in the areas they're being generated? So more crowded hospitals could have a higher morbidity and mortality independent of the impact of vaccines.
Yeah, it's a very good question, and it raises the issue of vaccine efficacy and vaccine effectiveness. And many of you will be familiar with those two terms because we think about this often with flu vaccines. So vaccine efficacy is we assess the effect of the vaccine on a predetermined primary endpoint in the setting of a clinical trial. And so that's what we have evaluated in all the studies together to date because they're placebo controlled. So it's exactly the same environment for the placebo, recipients of the placebo and the recipients of the vaccine, and you can get a very accurate number of vaccine efficacy. Vaccine effectiveness is when you put the vaccine in the field and you see how good it is at preventing or impacting the sorts of numbers you're talking about, such as admissions to hospital or mortality. And we will start to get an assessment of vaccine effectiveness, especially amongst countries that are rolling out this vaccine. Israel has already reported some preliminary findings on vaccine effectiveness after vaccinating millions of people in Israel. And those studies will emerge now that high numbers of people are being vaccinated around the world.
Okay, thank you. And Chung asks, do we know how long the vaccine will likely be protective after administration?
We don't know the answer to that. The longest published data that I have seen at the moment on antibodies, and if you assume that antibodies are the appropriate biomarker for protection, and we think that's the case, for the Moderna vaccine, they've followed people out for over 100 days now, and vaccine antibody levels, although dropping slightly, have actually been maintained over that duration of follow-up in all age groups. So we know to about 100 days. We don't yet know beyond that.
Okay, and Shide asks, can incomplete vaccination, like getting one shot of the vaccine instead of two, cause mutations in the virus?
There is some concern about that, or prolonging the time interval between two vaccines, although that has not been formally assessed, of course. What could drive new mutations? Subtherapeutic or lowish levels of an antibody where the virus can still replicate in the presence of that antibody. It's a nice way to think about this is how we think about drug resistance, which I think most people on the webinar would be familiar with. That if you have a suboptimal level of an antiviral drug and the virus is replicating in the face of that drug, it's a driver for the virus to develop resistance. We're very familiar with that in HIV, for example. And theoretically, that could happen after a dose of a vaccine that gives you a suboptimal level of antibody. Hasn't yet been proven. But it is theoretically possible. We know that the second the second vaccine dose for all the vaccines that have been developed to receive 2 doses, there is an increase in antibody level after that second dose. It's worth highlighting that the Johnson & Johnson vaccine, the results, which have only been released by press release that I talked about in the later part of the talk, is actually a vaccine that only has a single dose, but all others have been evaluated with 2 doses.
Okay, and Milagros asks, I have some patients that are in the first trimester of pregnancy. Is there enough evidence regarding efficacy or safety in pregnant women?
In all of the studies to date that have been published, the pregnant women were not included in those studies, and the recommendations are that it should be discussed at an individual level with each pregnant woman. It's very unlikely to have any adverse effects, and data is being collected now in the phase 4 of rollout of vaccine in pregnant women. I'm unaware of any data at the moment on safety or efficacy.
So that's all we have time for today. Thank you very much, Professor Lewin, and thanks to our listeners for joining us. As a reminder, to view the COVID-19 Comprehensive Resource Center on the Clinical Care Options website, click on the link in the show notes. Please be sure to check back regularly for more episodes on important infectious disease topics. Thanks!